Bismillah ar-Rahman ar-Rahim. Cell membrane is composed of a phospholipid bilayer. Glycoproteins and sphingolipids are the major components of the cell membranes. Now coming to the properties of the cell membrane. With regard to the physical and mechanical properties, the cell membrane can be described as a fluid mosaic model. The aqueous environment inside and outside of a cell prevents membrane lipids from escaping from the bilayer, but nothing stops these molecules from moving about and changing places with one another within the plane of the bilayer. The membrane, therefore, behaves as a two-dimensional fluid, which is crucial for membrane function. The fluidity of a lipid bilayer depends upon its composition. This property is distinct from membrane flexibility, which is the ability of the membrane to bend. The proteins are floating in the sea of membrane phospholipids. The distribution of proteins forms a mosaic-like pattern. The proteins can move laterally. The following factors affect the fluidity of the cell membranes. The hydrocarbon tails of phospholipids containing short chains make the cell membrane more fluid. Next is the degree of unsaturation. Kinks that are produced due to the presence of double bonds make it difficult for the tails to pack against one another. Larger the portion of unsaturated fatty acids, the larger the larger is the number of the kinks and more fluid is the membrane. And temperature increases the thermal motion of the phospholipids, so increases the fluidity. Cholesterol stiffens the cell membrane and makes it less fluid and less permeable because it fills the spaces between neighboring phospholipid molecules that are caused by the kinks in the unsaturated hydrocarbon chains. The interior of a lipid bilayer is normally highly fluid in the liquid crystal state. Hydrocarbon chains of phospholipids are disordered and in constant motion. However, at low temperature, a membrane containing a single phospholipid type undergoes transition to a crystalline state, in which fatty acid tails are fully extended, packing is highly ordered, and van der Waals forces between adjacent chains are maximal. So, kinks in fatty acid chains due to cis double bonds interfere with packing of lipids in the crystalline state and make the membrane fluid. Cell membranes are generally asymmetrical, presenting a very different face to the interior of the cell than to the exterior. The two halves of the bilayer often include uh, strikingly different selections of phospholipids and glycolipids. Moreover, the proteins are embedded in the bilayer with a specific orientation, which is crucial for their function. The lipid asymmetry begins at the point of manufacture. In cells, new phospholipid molecules are synthesized by the membrane-bound enzymes that choose substrate fatty acids available in one half of the bilayer, that is one monolayer, and release the newly made phospholipid into the same monolayer. To enable the membrane as a whole to grow, a portion of the lipid molecules then have to be transferred to the opposite monolayer. This transfer is catalyzed by enzymes called flipases. It has been suggested that flipases may transfer specific phospholipid molecules selectively so that different types become concentrated in the two halves of the monolayer. One-sided insertion and selective slipages are not the only ways producing asymmetry in the lipid bilayers. However, in particular, a different mechanism operates for glycolipids, which are a class of lipid molecules that show the most striking and consistent asymmetric distribution in animal cells. To explain their distribution, it is necessary to look more carefully at the route by which new membrane is produced in eukaryotic cells. Cholesterol, an important constituent of cell membranes, has a rigid ring system and a short branched hydrocarbon tail. Cholesterol is largely hydrophobic, but it has one polar group, a hydroxyl, making it amphipathic. Cholesterol inserts into bilayer membranes with its hydroxyl group oriented toward the aqueous phase and its hydrophobic ring system adjacent to fatty acid tails of phospholipids. The hydroxyl group of cholesterol forms hydrogen bonds with polar phospholipid head groups. Interaction with relatively rigid cholesterol decreases the mobility of the hydrocarbon tails of phospholipids, but the presence of cholesterol in a phospholipid membrane interferes with the close packing of fatty acid tails in the crystal state. Thus, it inhibits transition to the crystalline state. Phospholipid membranes with a high concentration of cholesterol have a fluidity intermediate between the liquid crystal and the crystalline states. Cholesterol is abundant in membranes such as plasma membrane that include many lipids with long chain saturated fatty acids. In the absence of cholesterol, such membranes would crystallize at physiologic temperatures. The inner mitochondrial membrane lacks cholesterol but includes many phospholipids whose fatty acids have one or more double bonds. So this is how that where there are unsaturated fatty acids, cholesterol makes it rigid, whereas where there are saturated fatty acids, the cholesterol makes them fluid by disrupting the closed packing, and this is the paradoxical action of the cholesterol on the membrane fluidity. Membrane fluidity enables the membrane proteins to diffuse rapidly in the plane of the bilayer and to interact with one another. It provides a means for distributing membrane lipids and proteins by diffusion from sites where they are inserted into the bilayer after their synthesis to other regions of the cell. It allows membranes to fuse with one another and mix their molecules, and it ensures that membrane molecules are distributed evenly between daughter cells when a cell divides. The cell membrane has resealing and bending properties. It grows and changes shape with the cell, enlarges its area by addition of new membranes without losing its continuity. It can deform without tearing. If it is pierced, it neither collapses like a balloon nor remains torn. Instead, it quickly reseals. Lipids in cell membrane undergo lateral movement, flexion or bending, and rotation. So, coming to the types of the membrane lipids. Membrane lipids are attached to glycerol backbone. The hydrophobic the hydrophobic tails are formed by fatty acyl chains, whereas polar hydrophilic head groups are formed by phosphate groups and alcohols along with any carbohydrate residue if attached. So membrane phospholipids are the most abundant membrane lipids. Different types of phospholipids are present in the inner and outer leaflets. The inner and outer leaflets are asymmetrical. Sphingomyelin and phosphatidylcholine are present in the outer leaflet, whereas phosphatidylserine, phosphatidylethanolamine, and phosphatidylinositol are present in the inner leaflet. Migration of phosphatidylserine to outer leaflet is a signal for cell death. Cholesterol is distributed equally in both leaflets. It can also flip-flop between inner and outer leaflets, whereas phospholipids show only lateral movement, but no jumping or flip-flopping between leaflets without any catalytic help from enzymes. Plasmalogen is another lipid of the membrane. In plasmalogens, the fatty acid at C1 is replaced by an unsaturated alkyl group attached by an ether instead of an ester linkage. So, these have Al rather than Yl at the end of their names. Platelet activating factor contains a saturated alkyl group at C1 in ether link and a fatty acid at C2. Platelet activating factor mediates inflammatory responses and platelet aggregation. It is synthesized in peroxisomes. Sphingolipids have alcohol sphingosine in their structure. Their synthesis requires vitamin B6 and B3. These are present in membranes of nerve tissue. Myelin sheath contains sphingomyelin with long chain fatty acids, whereas gray matter has sphingomyelin with primarily stearic acid. Ceramide is a membrane lipid. It serves as skin's water permeability barrier. The derivatives of ceramide include sphingomyelin and glycosphingolipids. So that's the sphingomyelin. It is present in nerve tissue. Next are the glycolipids. Glycolipids are the glycosphingolipids which contain a mono or oligosaccharide at their polar head group, and these play a crucial part in driving the lipid molecule to assemble into bilayers. These are always present in the outer leaflet, contain covalently attached oligosaccharides which are branched and smaller than gags. So that is a glycolipid. Glycocalyx is composed of carbohydrate moieties of membrane lipids and proteins. Acts as an ID tag that enables the body to determine self or antigen, whether an antigen is self or foreign. Now coming to the glycoproteins. Transmembrane proteins embedded in plasma membrane facilitate the transport of molecules across the plasma membranes. Glycoproteins contain the branched carbohydrate moieties attached to the proteins. These may be O-linked or N-linked. In case of O-linked, these are linked with the OH of the serine or threonine residue in the protein, and in case of N-linked, these are linked with the amide nitrogen of the asparagine residue. So that's an example of selectins that contain a carbohydrate domain called lectin on the extracellular side. The lectin containing domain of selectin on one cell interacts with the lectin containing domain on another cell, and it is important in immune responses and cell migration towards the sites of inflammation. Glycolipids are the lipids which covalently attach to oligosaccharides, and these form the basis of AB or blood group antigens. The simplest glycosphingolipids are cerebrosides, which then there and these are the ceramide monosaccharides, means that a monosaccharide is attached to a ceramide. Then there are ceramide oligosaccharides, which are produced by attaching additional monosaccharides, and gangliosides are derivatives of ceramide oligosaccharides, and they also contain one or more molecule of neuraminic acid. This is how glycosphingolipids are oriented in the outer leaflet of cell membrane. Glycocalyx cushions and protects the membranes physically and chemically, enables immune system to recognize and attack invaders, serves as defense against cancer. Changes in the glycocalyx of cancer cells allows immune cells to recognize and destroy them. It forms the basis of compatibility in organ and tissue transplants, plays part in cell adhesion and migration, causes neutrophils to adhere to blood vessels and then migrate to site of infection. It also guides embryonic cells to their destinations during development. Oligosaccharides and polysaccharides absorb water and make cell surfaces slimy. This helps in lubrication. Slimy surface of blood cells due to glycocalyx helps them to squeeze through narrow spaces. It helps in recognition of an egg by sperm cell and in recognition of invaders by immune cells. It also plays part in cell adhesion. Highly silated glycoproteins with very large mucin-like domains on cancer cells are present. These strongly reduce cellular adhesion. There is loss of adhesion and cells due to adhesion in the cells due to abnormal distribution of silated glycoproteins. The entire cell surface of cancer cells is covered with these silated glycoproteins, which helps in invasion and metastasis. Now coming to the membrane proteins. Membrane proteins make up 50% mass of the plasma membranes and perform different functions. For transmembrane proteins, the hydrophobic regions of the proteins come in contact with the hydrophobic fatty acid tails of the cell membrane. Peripheral proteins are present on the cytoplasmic surface by weak linkages. These can be washed using simple salt solutions. Peripheral proteins are on the membrane cytoplasmic surface. They are water soluble with mostly hydrophilic surfaces. Often peripheral proteins can be dislodged from the membranes by conditions that disrupt ionic and H bond interactions. For example, extraction with solutions containing high concentration of salts, change of pH, and or agents that bind divalent cations. This diagram shows peripheral membrane proteins like kinases present towards cytosolic surface and also the alpha, beta and gamma subunits of G protein coupled receptors, which also represent peripheral membrane proteins, the alpha, beta and the gamma subunits. Ankyrin are a family of adapter proteins. These are peripheral proteins that mediate the attachment of integral membrane proteins to spectrin actin-based membrane cytoskeleton. Spectrin and actin are peripheral membrane proteins. And chitins have binding sites for spectrin. The inner or cytosolic side of red blood cells membranes is shown here. The actin binding protein spectrin network is shown by brown color and actin filaments by purple. Integral proteins have domains that extend into the hydrocarbon core of the membrane. Often they span the bilayer. Intramembrane domains have largely hydrophobic surfaces that interact with the membrane lipids. The integral proteins have unique orientation. They form pores and they contain more than one membrane spanning alpha helixes or beta sheets curved into a cylinder or barrel. They may be having a lipid anchor anchored by covalent bonds. These can be removed by detergents. Some proteins are attached via GPI anchor, which is glycophosphatidylainositol. These proteins protect the cells from immune system and increase the lateral mobility. Proteins having lipid anchors may be present on the external surface via GPI or glycophosphatidylainositol, or these may be present on the cytosolic surface of the membrane. Here they may be linked to the isoprenoid group. So glycoproteins are present on the extracellular. Fibronectin is a glycoprotein of the extracellular matrix that binds to membrane spanning receptor called integrin. Fibronectin binds extracellular matrix components such as collagen, fibrin, and heparin sulfate proteoglycan. Membrane domains are found by different lipid composition between apical and basolateral membrane. Domains of epithelial cell plasma membranes have applications in endocytosis, membrane trafficking, and signal transduction. Membrane proteins can be tethered to cell cortex inside the cell, to extracellular matrix molecules outside the cell, to proteins on surface of another cell, and they also form diffusion barriers or tight junctions and can restrict the proteins to a particular membrane domain. Lipid rafts are formed by membrane glycosphingolipids, cholesterol, and GPI anchored proteins. These laterally mobile microdomains of plasma membrane regulate signaling and trafficking functions of the membrane. There are three types of lipid rafts: glycosphingolipid enriched membranes or GEMs, phosphoinositol rich rafts, and caveolae. The caveolae are flask shaped invaginations of the cell membrane containing protein caveolin.